Okay. So you guys online can see something. Okay, better? We're doing all right? Yes, it may have taken me half the day to get here, but I'm here. That's the important thing, right? No, right. What do they always say? Showing up is half the battle. Well, not really, not if the battle's already over. It's a shame. Okay, so one of the things I wanted to do is I wanted to cover, and I said I wanted to take a look at what Mendel did, and I wanted to see it through his eyes. And the other thing I wanted to do is I wanted to kind of calibrate this to scientific method. Now, what he did was monohybrid cross and dihybrid cross. So the first step in the monohybrid cross is we're going to do what T.A. Knight did, which was take your true breeders. In this case, he took a true breeder, and where he had like 7 different traits that he was looking at. We're just going to pick flower color since I got the purple pen up here. And so basically what he did is he took a true breeder for purple. And he crossed it to a true breeder for white. Now, I don't you to cheat ahead. I don't want you to start laying genetics on there like we've already done with the genetics lab, right? Talking about dominance and that sort of stuff. I want us to tease this out, and I want us to basically pull out and sort of reason our way through Mendel's data and to show you how with just a little bit of logic and rationale, you can see how it would be fairly easy that you come up with the same conclusions, right? Now, first of all, I want to correlate this with scientific method. And the reason why I do that is because, of course, I've got a bias for genetics, right? So basically all the world revolves around genetics, which is not just bias, it's actually true. But the scientific method, we all kind of learn about in the first chapter, and we kind of know all the different parts and pieces, and we think, oh, well, this is how you do scientific research. But in reality, that's not the way we did it before this. I mean, there were pieces of scientific method that were kind of coming together, but there wasn't a logical stepwise progression. And nobody had really done it before. I mean, even Darwin himself, even T.A. Knight, I mean, they didn't really fully unfurl scientific methodology the way we know it now. The first time that we've seen that is actually what Mendel did. In a lot of ways, I like to say that Mendel didn't just kickstart genetics. Mendel actually taught us how to do science. He is probably the clearest, the most discrete illustration of how to implement the scientific method to do research. And as his example, I think kind of sets us up for the 20th century. And so in a lot of ways, all the rigor and the reason that is associated with scientific research in the 20th century, in large part, has a lot to thank Mendel for, because Mendel kind of showed us how to do it. Okay. Now, it's a very biased way to look at it, but it's true. So what are the pieces? First, observation, right? And we're going to correlate to how exactly, as Mendel goes through his experiments, how he basically hits on every single piece of the scientific method. Then after that, there's a question. And then your best answer, not the correct answer, but your best guess, is your hypothesis. And then from your hypothesis, you should be able to generate a couple of if-then statements, right? Those are your predictions. And then from that, you're going to do an experiment. You're going to collect data. You're going to analyze it. And then you're going to come up with some sort of a conclusion. That's your basic 6-step scientific method. Mendel hits on every single one. Most research that we've done in the 20th century hits on maybe a handful of them, a few of them, but not necessarily all of them. So let's go ahead and take a look at his first step. So he first sets up the parental generation, right, which is crossing a purple true breeder with a white true breeder. And then what does he see in the second generation, his first filial generation? He sees all purple. Okay, here's the problem. Let's make an observation. What's our observation? Our observation, it's all purple. Right, F1s that is. That's your observation. What's your question? What's the screaming question here? The most obvious one. Yeah, exactly. That's your glaring question. What happened to the white? So that's your question. So notice step 2. Okay, now what's your best guess? Remember, there's 2 hypotheses that are rattling around at this particular time. One is blending and the other one is particulate. So according to the blenders, they would say basically that it's gone, right? Because once you mix blue and yellow are gone. Everything's green from then on. So basically they're going to say it's gone. But here's the white is gone. That's what the blenders say. What do the particulate guys say? It's not gone. White must be hiding. Right? What is that? That's your hypothesis. Now you have some predictions that come from your hypothesis, don't you? If 1 is true, right? So for number 1, let's come down to this testable prediction. If gone, then what's going to happen? It's non-recoverable, right? You're never going to see it again. Guess what that is? It's an if-then statement. It's a prediction. For the second one, if it's hiding, then it should be recoverable. That's your prediction for number 2, the if-then statement. So he's already satisfied the first 4 of the scientific method. Now what does he do? Now he conducts an experiment. What if I cross the F1 purples to the F1 purples? What am I going to get? What does he see? He sees 3 purple to 1 white. So he did an experiment, right? Drew some data. So what are his conclusions? His conclusion is the white is not gone. Remember, your conclusion has to answer your question. Your question was, where's white? So basically what he did is he proved that white's not gone. Why? Because in the F2s, I get it all back. Okay, so this is, and so that's basically, you can see how basically he just pretty much used all 6 steps of the scientific method. Very clear, very simple, and he got a lot of good data. So now you're probably thinking of this, and you're probably thinking, okay, that's cute, right? I mean, 3 to 1, but we counted corn, and I'm pretty much an amateur. So how come Mendel gets into textbooks and I don't? Right? So it's kind of cute. So he finds a 3 to 1, but didn't you guys count up a 3 to 1? So how come he gets the textbook and you guys don't? Because he did more than just this. All right, let's reason through this. What I want to do is I want to build a glossary of some ideas, and I want to put them all in one place. And I am jumping the gun on some of these slides, but I don't really care because I want it more all in one place. I want us to sort of reason our way through this. There's a couple of questions here, first of all, that we are looking at. I'm going to call it a genetics ideas. The first one that we can get from our data is that there is like a packet of instructions. It's a particulate. This suggests a particulate inheritance. Why? Because your conclusion basically was not consistent with blending, right? So what this suggests then is that you have what we now call a gene, right? This is your particle. So what Mendel observed as a particle of inheritance is what we now refer to as a gene. But guess what? Mendel also noticed a couple of other things about this, right? Because he was very observant. He also noticed that in this particular case, my gene, which in this case governs flower color, that's what we're working with, has two different versions to it, right? You have a purple flower color version and you have a white flower color version. So before we even get started, we've already kind of laid some of the groundwork, right? These versions of the same gene, like for instance, is purple flower color? Is it governed by the flower color gene? Sure. Is white a flower color? Is it governed by the flower color gene? Okay, so we don't have any argument that this is exactly the same gene, right? But what we have is two different versions of it, a plan A and a plan B. So these two different versions we later on called alleles. Okay, doing good now, right? But wait, let's look a little deeper. Let's take a look at our data. So we cross a true breeder for purple with a true breeder for white, and what do we get in F1? All purple. Well, first of all, according to our data, our conclusion has already been that white is not gone, right? That means it's there somewhere. So the question is, how come you can't see it in F1? So basically, if you're saying that you have a purple packet of instructions from the purple parent, and a white packet from the white parent, then that means the F1s have to have one of each. That means they must have both versions in their hands. But which one do you see? Purple, right? So basically they're saying that white is hiding. So what they said then basically is that these alleles, these multiple versions, don't have the same strength to them. That one allele is dominant. So a dominant allele expresses its phenotype in one copy. Now I'm kind of defining a definition with a definition there, but we'll define the definition a little bit later. Okay. He also noticed that white, being the other allele, is not seen at all, which means it must be weaker, capable of being covered up. This one then becomes the recessive. This one expresses in two copies and is masked by a dominant. So now you have this idea that alleles aren't necessarily the same, that you have strong ones and you have weak ones. The strong ones will cover up the weak ones, so you never see them. They're hidden. Okay, let's dig a little deeper. ask you a question. If I were to show you this flower right here, right next to this flower right here, would you be able to tell me which one is which? Is the correct answer yes? Well, think about it. What color are they? If I were to take the two of them, do the shell game thing, and say, tell me which one's the parental and which one's the F1. Could you do that? No, because they're both purple, right? So they both look the same. However, do they behave the same genetically? Let's take a look at the parental. The parental genetically behaves as what? A true breeder, right? Does F1 act like a true breeder? No, because it's got white in there, right? So do they behave the same genetically? No, they look the same, but genetically they behave differently. One's a true breeder, the other one is not. So what Mendel basically was fleshing out was the difference between phenotype, which is the way an organism looks, and genotype, which is your genetic makeup at a particular gene. You can think of that as your allele makeup. Now, normally, we like to think that the relationship between genotype and phenotype is 1-to-1. And in the Mendel situation, it is. But in most other situations, it isn't, right? That your phenotype, the way you look, is actually a complex of genotypic things, that is genetic things, right? Allele things, plus non-genetic things. And there's a lot of that in there. Some random things that are in development, and of course, there's also some environmental things. And there's a lot of different variables that actually goes into developing your phenotype. A good chunk of it is genetics, but there's also a pretty hefty chunk that's non-genetic. And those are all stories and discussions that we flesh out more. We have time to flesh out more in the genetics class, which I know you're all taking. Okay. So then what kinds of genotypes do we have then? Well, this kind of becomes the next obvious thing, right? So basically, if these guys are genetically different, right? They're genotypically different, then what does that look like? Well, think of it. First of all, a true breeder has only instructions for purple, right? So whatever purple is, that's all it's got. Now, in this particular case, and this kind of came along at the turn of the 20th century, we first of all know a couple things about purple. Number one, it is the dominant allele, right? And number two, white is the recessive allele. The other thing that we also know is that if you're a true breeder, what that basically means is you have only purple instructions, which means that you have two copies of the dominant allele. Now, notationally, what we always do is we use an uppercase letter for dominance. So this basically we said is going to be dominant, big P. And since it only has instructions for purple, it's going to be big P, big P. The white, on the other hand, has only instructions for white, so it'll have little p, little p. So then, when the purple plant puts its gametes forward, it only has a big P to give. The white parent only has a little p to give. Same thing with this one here. So then when you look at it, what you see are three different genetic makeups. First one is called the homozygous dominant. here you have 2 copies of the dominant allele. So for instance, big A, that's your homozygous dominant. Then you have another homozygous recessive, where here you have 2 copies of the recessive allele, which is little a. And the last one is what's called a heterozygote or heterozygous. This is where you have 1 dominant allele and 1 recessive allele, or big A, little a. So these are your 3 dominant genotypes. But let's take a look at the consequences of these, right? So here's our genotypes: homozygous dominant, homozygous recessive, heterozygous. But what would be the phenotypes for this? Well, the phenotype for this one would be whatever the dominant phenotype is, right? In the recessive, it's going to be the recessive phenotype, right? Because you have 2 recessives. And in the heterozygote, because you've got that 1 dominant masking the recessive, this is also going to look like the dominant phenotype. So you can see that there's a little bit of a disconnect between genotype and phenotype. So what else did he do? Well, basically, this is kind of what Mendel did. So you can see here, you've got your parental generation, which is going to lead to your F1s, which is going to lead to your 3-to-1 here. But this is what's called your phenotypic ratio. There's also a genotypic ratio, right? That is how you behave genetically. So you already know that you have a phenotypic ratio of 3-to-1, right? 3 purples to 1 white. But now the question is, what are you genotypically? Well, what he did was he took these guys here and selfed them. And what he noticed was that these guys were true breeders for purple. Then he took this guy and selfed it, and this was a 3-to-1. Took this one and selfed it. This was a 3-to-1. He took this one and selfed it, and this was a true breeder. So what he had was 3 major genotypic classes: 2 true breeders and 1 big group of non-true breeders. It turned out that 1 was a true breeder for purple, 2 were non-true breeders, and 1 was a true breeder for white. This then becomes what's referred to as the genotypic ratio. So Mendel saw a phenotypic ratio that was different than the genotypic ratio. So for every 1 homozygous dominant, which is all purple, true breeder, you get 2 heterozygotes, which are dominant phenotype, but non-true breeders, right? And then 1 homozygous recessive, which is phenotypically white, but a true breeder for white. Make sense? So that right there is actually a lot. So what Mendel basically did was basically show us in this one experiment how to execute and implement the scientific method. He also basically developed all of the answers. He developed a very, very handsome glossary in genetics for us to use for the rest of the century. Now, you actually, if you want to, it's kind of cool. You can actually read Mendel's paper. It's still out there. I've actually got it as part of my course shelf for the genetics course. It's a lot of fun to read because what you see in Mendel, I'm going to sit down because I've got like a hip flexor issue here. It's not working for me when I'm standing. But basically what Mendel did is he didn't come up with words like homozygous dominant. Remember, when Mendel was doing his work, they didn't know anything about cell stuff. They didn't know anything about mitosis. They didn't know about meiosis. They didn't even know about chromosomes really when Mendel did his work. So he didn't know any of that stuff. All that stuff came later. So what he was looking at was flow of information. Matter of fact, we now know that genes are on chromosomes, but that took a good 20 years in the first 20 years of the 20th century to really fuse those two together. The way it started was initially you had people over here studying chromosomes with like microscopy and stuff like that. And then people over here just studying information flow. And that's what Mendel was doing. He was just studying the flow of information in organisms from one generation to the next. That's basically what he was doing. So he didn't come up with these terms. These terms all came later. Now, what's fascinating about Mendel's paper is you don't see the terms in there. What you see is the definitions. So Mendel's describing something. It's like, okay, what he's describing is basically an allele, right? Or what he's describing is essentially a dominant recessive relationship. So he's kind of like describing, like theoretically in his mind, what we later basically put labels to. And we learn the labels now in class. It's also fascinating too. It's a fascinating read because in his paper, you can actually see the kernels of a lot of other branches of genetics. You can see the kernels of the statistical nature of genetics starting to form. He kind of lays that down there, doesn't get into it because statistics was not well formed at that point, right? But he kind of lays down some basic ideas that basically lead in that direction. The other the cool one is population genetics, like Hardy-Weinberg. Hardy-Weinberg equilibrium is a population genetics calculation that we use to sort of characterize the stability of gene flow within a population of organisms as they're sort of mating, and how different genes are kind of segregating and sort of getting fixed into a population of species. And we got this quadratic equation basically is what it is. You never thought algebra would come in useful. Well, here it is in population genetics, right? Right. So and you can see the beginnings of that quadratic formula actually being discussed in Mendel's paper. So there's like a little kernel of population genetics, and population genetics comes well after a lot of the more core genetic stuff. So it's really fascinating to read his paper because you can see now, kind of being on this side of genetics, you can look back and see like, oh yeah, I can see the beginnings of that field of genetics happening, and that one, I can see that one happening. And so it's really fascinating because he really did kind of set up the discipline for all these different avenues of genetics. That's the reason why he is probably the greatest name in the book, the textbook, that is. Okay. So what are his conclusions? Basically, he kind of pinned down the dominance recessive relationship. That's one of the things that he pinned down. And he also really looked at segregation, right? Because there's a couple of things that Mendel's really known for. Three things, right? First of all, discovering his 3-to-1 relationship, right? That was a big one that he's known for. And his ratios. Actually, today in genetics, that's one of the ways we actually determine how a gene is genetically behaving is by these ratios, very similar to the way Mendel approached it. We do the same thing now in genetics as well. Like even in human genetics, right? We basically get somebody's family pedigree. We kind of chart their pedigree. Did Uncle Fester have it? Yes or no? Did Aunt Margaret have it? Yes or no? We kind of fill out this huge pedigree and we try to look for these patterns like this. And we look for ratios. Well, it's like if it's a recessive disorder that's going through your family, then we should expect this kind of ratio, right? And so that's actually, to this day, we take the exact same approach that Mendel took in the 19th century. Now, what Mendel came out with was a couple of major things. Obviously, a big takeaway was that genetics was driven by discrete factors. In a way, this was sort of like the end of blending. So just like Pasteur basically signaled the end of spontaneous generation with the development of sterile technique and the construction of microbiology, when Mendel did this, it basically signaled the end of blending. Because you can't both blend and be particulate at the same time. The other big one was that you only get one copy of the gene from each parent. It seems kind of like a yeah, duh moment for us now, but that was a big deal back then. We didn't really know that for sure. And now we do. The idea of the allele, we didn't know that before either, right? That there's alternative versions of a gene, and this whole concept of genotype and phenotype, right? So this whole homozygosity, heterozygosity, we didn't know any of that either. So there's like a ton of stuff that actually Mendel discovered for us and really set us up nicely. So we already talked a little bit about dominance and recessiveness, our genotype and phenotype. So we won't spend a whole lot of time there. But the big thing, one of the big things that Mendel's known for, there's two laws of genetics or principles of genetics, doesn't matter. It's the same thing. Mendel's known for. The first one is segregation. Basically, what he's saying is that you have two alleles that any individual will get. You're going to get them during gamete formation. Now, here's the problem. Mendel didn't know anything about gamete formation. He didn't know about meiosis. You know now more. You know more now than Mendel knew. He didn't know anything about meiosis or gamete formation. He didn't know about random fertilization. He didn't know about recombination. He didn't know any of that. What he saw was that there were two distinct versions of this allele that were being segregated in each of the parents' gametes. What he knew was that when he took that pollen with his little paintbrush, he knew that that was the male sex cell, right? That was basically the male's sperm, essentially. And when he brushed that on the stigma, he knew that that was the female's sex cell. And so if you have the male with one packet of instructions, adding his to the female who has another packet of instructions, then they could have only come from those two sources, right? So the idea that we only have two alleles, one from each parent during gamete formation, is the principle of segregation. That's how we understand the principle of segregation now. This is, by the way, also one of the reasons why he did reciprocal crosses. Because if you're going to say that my purple male to my white female, basically my male is donating the purple instructions, then you'd better have consistent results if you were to flip that script, right? So if you go white female, purple male, then you should see a consistent pattern of data, right? This suggests that you're getting one from each parent. In this case, you're getting white from the female and purple from the male, or vice versa. That's the reason why he did reciprocal crosses, is because for the testing of this principle of segregation and coming through parents. Now, later on, now here's what happened. That was really cool work. And if you read Mendel's paper, it is pretty cool, if I don't say so myself. But here's the problem. That was like around, what, 1865 or so? The world was kind of in a bit of a state there. That wasn't really all that long, maybe 15, 16 years post Darwin's paper. So there was a lot of uproar over the whole evolution thing. So if you study scientific history, you know that discussion got rather heated from a lot of different camps. So when Darwin published that paper, it created a lot of, what do I do with this kind of moments in a lot of areas, in a lot of disciplines, right? So this is a paper and a concept view, basically presented by science, that kind of forced the other two major intellectual disciplines to sort of figure out what to do with it, right? And so there was a lot of that going on, and there was a lot of heated arguments. This, by the way, if you're curious, is when the whole discussion, and which this is basically the era when, unfortunately, evolution got tied up into a theological debate between atheists and theists, right? Which kind of led, I would argue, evolution kind of off the rails a little bit in the weed patch a little bit. But that's kind of where a lot of this came in. Why? Because we didn't have the genetics to talk about evolution properly. So all the arguments for evolution were basically the rock zoo, right? So, you know, just stuff like that, right? Stuff that wasn't really all that satisfying. And so there was a lot of angst and anger and back and forth about it because we didn't have the genetics to view it properly. And so that's that was going on now, right? So we're talking about 16 years now post Darwin. So that was leading the charge. That was like kind of what the world was about. By the way, in the US, we didn't really care so much about that. We were basically licking our wounds from the Civil War in 1865. We just lost our president. At this particular point, we had the stooge Johnson, not the second one, but the first one, the one who actually got impeached, the first president to get impeached. Almost, he had to actually resign. So this was Andrew Johnson, not Lyndon Johnson. So turned out to be probably one of the worst presidents in the country, in the history of the country. So this is where we are, right? We're post-Civil War. The North and the South are basically trying to figure out how to coexist now after this big bloody battle. We got Reconstruction happening on. We're post-Emancipation Proclamation. So we've got society is trying to rewrite the rules and the lines. Their first foray beyond sort of white male dominated thinking and getting into, electing Black congressmen, which actually happened in those early days after the Civil War. So this is what's happening in the US. It's a very, very difficult time in the US. We're really kind of going through an identity crisis, and we're trying to sort of forge our identity. So this got lost. So the whole story is the world was doing other things, and Mendel's work got lost for a good 4 decades. And then 4 decades later, around the turn of the century, then it got rediscovered by a group of geneticists. William Bateson is one of them. William Punnett is another one. And what they did is they took Mendel's paper, read it, were like, hey, this is great stuff. And then they did what every scientist does. So like, listen, does what Mendel's CMP plants, is that going to work for my system? Like, I work with corn. So if I do the exact same experiment that Mendel does, will I see the exact same results that Mendel saw? Or like, I work in 4 o'clock plants or whatever, right? So that was basically what they did. They're also the ones who came up with the words, the terms, right? So they put gene on the particulate piece. They put allele on there. They put homozygous dominant. So all these words were inventions of this generation of geneticists. They also created what's called the Punnett square, which is basically an organizational tool used to basically predict outcomes. For instance, if you take a look at this is your Punnett square. Up here, you're going to put your gametes for males, and then down here is going to be the gametes for females. And then when you take a look at, say, for instance, this individual here, they can only produce 2 types of gametes. They only produce a large P gamete or a little p gamete. That's it. Same thing's true for this individual over here. They're either going to produce the large P gamete or the little p gamete. So when you sort of organize this into sort of the square, then what you do is you just basically pair these up. So what if this pollen actually fertilizes this egg? Well, that means I'm going to get an individual that's big P, big P. Then what if this pollen fertilizes this egg? Then I'm going to get a big P, little p. And if this pollen fertilizes this egg, I'm going to get a big P, little p. And if this pollen fertilizes this egg, I'm going to get a little p, little p. That's basically what's happening here. Okay. So you're kind of filling in your square by pairing these potential matings up. Now, if you're a piece of corn, then you will see all of these possibilities. But not if you're us. If you're us, because we only have 1 kid at a time, you're going to see 1 of these 4 possibilities. Okay. Now, if we had more, then you should see, if you have enough kids, you should see all 4 possibilities. So that's what the Punnett square does. So the nice thing about this is it's going to allow you to basically plan out a cross and the outcomes of the cross before you actually even set the cross up. So let's apply this to Mendel's system. So what he saw was a true breeder for purple, which means you had big P, little p for white. When you cross them together, all you got were heterozygotes, right? That's your F1 generation. Then once you broke up and you crossed your F1 generation to each other, what you had was a big P with a little p, and then another big P with a little p, and then what you got was your homozygous dominant. This gives you your heterozygous. This gives you your heterozygous. This gives you your homozygous recessive. So in this case, you can see you've got two heterozygotes, one purple, one white, for your 3-to-1 phenotypic ratio and your 1 to 2-to-1 genotypic ratio. You can see you can use the Punnett square to organize your alleles before you even cross the first flower. As a matter of fact, this is what we do in genetics. If you recall, I mentioned that I was at UC Davis working in a genetics lab on flies when I first started thinking about that story of Jacob that I started off with, right? And this is exactly what I was doing. So there was a lot of crosses that I wanted to do, and it's what we call a mating scheme in genetics. So you kind of create this big mating scheme. And so what you do is you take one genotype of fly and you cross it to another, and then you basically draw out all the genetic outcomes and say, well, I should be able to get this many of this, and this many of this, and this many of this. And then you pick your exceptional female, right? That's the little white lamb in the story. And then I'm going to mate this particular individual with this genotype, and I'm going to get these potential outcomes, and I should get this number or this proportion of each of these outcomes. And so then I'm going to pick this. And so you have this big elaborate mating scheme until you get to finally the ultimate data that you wanted. And you did all that on paper before you even set up the first fly. And then what you do is you basically go back, you set up all these flies, and guess what? The crosses do exactly the way you mapped it out. Okay. The reason why that's important isn't because, that's kind of all cool and I want you to know what happened in a fly lab. But the reason why that's important is because what you're doing is you're using this, what we now refer to, at this particular point in the 1900s, right? You're looking at it as the allele theory of genes, right? You don't know that this is true just yet. But one of the things you're able to do is use this sort of mating scheme approach to essentially predict what's going to happen in real life. Generally speaking, when you're theoretical, what you've written on paper predicts your actual, which is what you do in real life, then that is good evidence that you've got it right. Okay. So when you write out these mating schemes on paper, it is predictive of actually what's going to happen when you get in the garden and you start planting pea seeds. That typically means that you've got it spot on right. Okay. So now let's look at the next big thing that Mendel did. Let's take a look at the dihybrid, shall we? And the dihybrid, now you're kind of using the same thing. Only now instead of using one trait, you're using two traits. And here they use R and Y, and they do the same thing. So you use true breeders in both cases, but they're going to kind of give you this whole thing into sort of, you get your true breeders, which are going to give you your heterozygotes. You cross those together, and instead of a 3-to-1, it's going to give you a 9-3-3-1. So what I want to do is I want to take a look at this, and I want to show you how you get to this. So this is basically what they have, but what I want to do is I want to simplify this a bit. And let's go and drop it right in here. I'm going to do another slide. And we're going to do the dihybrid cross. So let me define a couple of terms. Oftentimes in genetics, you always define your terms so that anybody looking at your notes can know exactly what you mean. Instead of using P and Q and other things, I'm going to use A and B because I can differentiate upper and lowercase easily. So basically, we have a gene where A is essentially the dominant A phenotype, right? And you have a little a, which is your recessive A phenotype. And then B is going to be a different gene, but this is going to be your dominant B phenotype. And you have little b, which is going to be your recessive B phenotype. So here's what I'm going to do. I'm going to take, and first of all, I'm going to create a true breeder cross, a parental cross. So I'm going to take big A, big A, big B, big B. So it's a true breeder for dominant for both genes. Now, one thing to remind you of, these genes are independent of each other, right? So I could equally write a true breeder as either big A, little b, right? That's still a true breeder. It's a true breeder for dominant for A, but true breeder for recessive for B. Or I can write little a, big B, big B. All three of those are true breeders. I'm just making them double dominant for sake of clarity, okay? And I'm going to cross this to a double recessive. That's my other parent, my other true breeder parent. Now, when I cross this, I'm going to get in my F1, I'm going to get a big A from one parent, little a from the other. And I'm going to get a big B from one parent and a little b from the other. This is what's called a compound heterozygote. I'm heterozygous at both genes, A and B. Notice, what's my phenotype now? It's dominant for both genes, right? Because they're both heterozygous. So let's go ahead and self these two or cross them, doesn't matter. So now what I want to do is I want to basically build a Punnett square because I've got a lot of possibilities here, right? First thing I need to do with the Punnett square is I need to define which gametes, like how many different types of gametes can each one of these individuals make. So let's first start off with this one. Remember, the A gene has to give an A allele, but it doesn't have to give a specific one. It can give either big A or little a, right? It doesn't matter. But it also has to package a B allele into the same gamete. So what happens then if I package a big A from the A gamete? Well, I have two different options here. I can either choose a big B to go with it. That's going to be one gamete. Or I can choose a little b to go with it. That's another gamete. What if I, however, choose little a instead? If I choose little a instead, I can either package it with a big B, that's another gamete, or I can package little a with a little b, that's another gamete. The same thing is happening with this individual here, and I'm going to put this individual down on the side. So they have a big A, big B, big A, little b, a little a, big B, and a little b. Now I can draw my Punnett square. Now let's go ahead and mate them up. I have this gamete with this one. We got big A, big A, big B, big B. This one's going to be big A, big B, big A, big B, little b, big A, little a, big B, big B, big A, little a, big B, little b, big A, big A, big B, little b, big A, big A, little b, little b, big A, little a, big B, little b, big A, little a, little b, little b, big A, little a, big B, big B, big A, little a, big B, little b, little a, big B, big B, little a, big B, little b, big A, little a, big B, little b, big A, little a, little b, little b, little a, big B, little b, little a, little b, little b. Easy, right? Okay, so let's go ahead and count phenotypes now, not genotypes. Okay, these are all of our products. Now, if we're working with corn, we're going to see all of these, right? Or an animal that has big litters, we're going to see all of these if we have enough offspring. If you're a human, you're only going to see one of these, typically, unless you have twins, right? Okay, so let's go ahead and count this. This one right here, what's the phenotype of that one? Dominant for both genes, right? Double dominant. So this is going to be a dominant. I have one of those. Okay, how about this one right here? Still double dominant, right? How about this one right here? Still going, right? How about this one right here? Another one, right? How about this one right here? Still there, getting bored, right? How about this one right here? So now you got a different class, right? This is a different phenotypic class. You got a dominant and a recessive. So dominant in A, recessive in B. So there's one there. How about this one right here? So double dominant, right? That would be a class 1. How about this one right here? So that would be class 2, right? How about this one down here? Third row. So that'd be class 1. Next one next to it. So that's another class 1. This one. So this is different, right? So we got something new. So this is recessive in A, dominant in B. So this is this one. Got one of those. So that's class 3 now. How about this one right here? So that'd be a class 3, right? That'd be this one. How about this guy down here at the bottom? So that'd be a class 1, right next to him. So class 2, guy next to that. So that'd be a class 3. And the last one, double recessive. So that's something new, right? What's my phenotypic ratio? Nine, 3, 3, 1. This ratio, just like 3 to 1 and 1 to 2 to 1, are diagnostic for dihybrid cross. However, this 933 only happens if both genes are randomly segregated. It's to say that what's happening at A has no bearing on what's happening at B. It's like flipping a quarter, right? If I flip a quarter with my right hand and then with my left hand, does flipping the quarter in my right hand have anything or any bearing on what happens in my left? They are two separate events, right? That's what we mean. That's what's happening with these genes, that they're completely random and that they're independent events. That's the only way you're going to get a 9331. So that's what Mendel basically saw. Now here's the question. Are all genes random like this? Do they all see a 9331? Well, of course not, right? Because otherwise there wouldn't be a genetics class. This would be the end of it. As it turns out, there's a lot of genes that are not random. A lot of genes where the events that happen in A do have an effect on the events that happen in B. When that happens, it changes the ratios. It changes this ratio. But the cool thing about it is the ratio is always founded on 9331. So here's a good example. One particular type of interaction between genes where they kind of mess with each other is called epistasis. And in that case, epistasis, there's a dominant epistasis, recessive epistasis. I'm thinking recessive. The ratio for that is 9-7-4. Or excuse me, 9-7, sorry. And so that basically is 9, but the other 3 are all the same. So it's like 3 + 3 + 1. So even like the epistasis ratio, which is 9-7, is actually founded on the 9331. So you also get like, 12-3-1, you can get 15-1, so you can get all different ratios. And in genetics, what we oftentimes will do is we will basically take a look at some sort of a gene. We will collect the data like this on its ratio. We'll figure out its ratio, and then we'll try to diagnose which which version of that it's segregating with. Okay. So that's your dihybrid cross. So in this case, when you're working with round yellow versus wrinkled, round wrinkled versus yellow green, then your double dominant is going to be your round yellow. Then your dominant recessive will be round green, and then recessive dominant will be wrinkled yellow, and then double recessive will be wrinkled green. Okay, so it always comes out that way, which is the reason why I wrote it as double dominant, dominant recessive, recessive dominant, and double recessive, because regardless of what the markers are, it will always be that. That makes sense? And that's kind of the way we approach it in genetics. It kind of makes it a little bit easier to deal with, actually, and to understand better. So this is kind of what it looks like. So here you can kind of see you've got your segregation. So at the top, you see your parental generation, which basically is segregating out. And here they basically put it on chromosomes. I mean, at this particular point, we know that a gene inhabits a location on a chromosome here, right? So you can kind of see how you're segregating out. These are the gametes here. But you see you're segregating out basically your different chromosomes. And so the traits that you're getting is actually information based on the chromosome. The reason why I don't like this, and I understand why they're doing it, is that they basically make it sound like with its proximity, like this is what Punnett and Bateson came up with. Like they're like talking, they're already following chromosomes and things like that. They're not, right? So what this is basically is a very, very strong sort of a backward view look of like, okay, now that we just mentioned Punnett squares, let's fast forward to the end of the 20th century and then look back, and now we can see that everything is tethered to a chromosome, right? And that's important, but that took 20 years to come together in genetics. So to kind of go from Bateson and Punnett to chromosomes is a little bit artificial. It's a little confusing, actually. But we all know now, because of molecular genetics, which didn't really bloom until like the 50s and 60s, that basically what you've got is DNA. What gene now is basically a sequence of DNA that's hidden or found on a particular location in a particular chromosome, right, called a locus. So each gene or each allele is basically located in a very specific region of a chromosome itself. And it's the chromosomes themselves that we're actually inheriting from our parents, right? So that's that actual discrete particle. So Mendel was right. It was an actual discrete particle that we're inheriting, even though technically speaking, he was thinking of more just the flow of information. And so here's your Punnett square. This is basically the same grid that I wrote out. It's a little more confusing because you got all these chromosomes, but you can kind of see how basically what you have is inside the Punnett square, you've got basically the allelic notation that we can sort of flesh it all out. And then you can actually see the discrete chromosomes as they basically come together. Okay, so let's talk statistics, shall we? Like, do we have to? Yes, we do. But light statistics. It's not going to be like Jeff Spence and going through an entire statistics course, although some of you do need to take biostatistics. So get comfortable with it. Okay, so here's the deal, right? So what Mendel basically saw was a ratio that can only happen if these two events are completely random. So what does that mean? Well, what that basically means then is that there's a couple of statistics rules that apply to how these are behaving. And there's only two of them that we really look at. The first one is rule of addition. The other one's rule of multiplication. And so this kind of gets us to our probability, right? Because anytime you have random events happening, probability and statistics is always going to be a part of the situation. So let's take first of all a look at the rule of addition. Now, the rule of addition is basically a situation where you want a particular event to happen in or a particular outcome to happen in either one event or a second event. So there's like multiple events. And so, for instance, you just want it to happen in one or the other. Now, the important thing about probability, especially the rule of addition, is that the rule of addition is a summing rule. So here's what it says. I want an outcome in event A to be a particular outcome. I want it to either happen in A or in B. A good example is flipping a quarter, right? So I'm going to flip a quarter twice. That's event A and event B. I want a heads in either A or B. I just want a heads once. I don't care when it happens. It can happen in A It can happen in B. I don't really care. I just want it to happen in either A or B. Or if you recall from your word problem dissections, or is the word trigger for summation. Right, we all remember learning that in school when we're diagnosing word problems. Anytime you see or, it basically means that it's an addition operation. So in this case, if you want it from A or B, then you add their individual probabilities. So let's take a look at this. So basically, we're going to do the cross of our two heterozygotes. And what we want to know is what is the probability of producing the heterozygote, right? That is that 2 class of the 1 to 2 to 1 genotypic class. Well, the probability of obtaining the first one is 1/4. How do you know that? Well, this is the reason why. Because this parent gave a big P. What's the probability of getting a big P from that parent? 1 out of 2, right? It's a 50-50. It's a heads, big P, or a tails, little p. So your chances of getting a big P from that parent is 1/2, right? So what's the chances of getting a little p from the other parent? Also 50-50, right? So here you basically have a 50-50 shot, which is where you get the probability of forming both of those together is going to be 1/4. However, the probability of getting little p from the first parent is still 1/2, right? And a big P from the other parent is 1/2, again, 1/4. So what you're asking for is what is the probability of getting a little p from mom or dad? That's what you're asking. And not a homozygous, right? But a heterozygous, a little p heterozygous from mom, or there's our trigger, or dad. Well, basically you take the probabilities of the individuals, which is 1/4 plus 1/4, which basically means you've got a 1/2 chance. By the way, you can also see this in the Punnett square, can't you? So what's the probability of getting homozygosity? Well, you have 2 out of 4, right? Which is what? 1/2. So you can see the Punnett square can break it down for you as well. Now, in this particular case, we're just asking, if I get that heterozygote from either mom or from dad, right? So it's an either or situation. Now, because of the dihybrid cross, this brought up Mendel's second big law of genetics, or principle of genetics, which is the law of independent assortment. And so basically what this meant is that each gene is sorts independently. Basically, it's random segregation. If it weren't random, you would get a different ratio, right? Just like what I was talking about with those other ratios, like 9:7 versus 12:3:1. Those are non-random segregations. We now know, of course, that there is the independent alignment of the homologs during metaphase 1. So we learned about that later, right? Mendel didn't know anything about that. All he saw was basically what looked like a pattern that is associated with the random segregation of 2 genes that had nothing to do with each other, right? So that's his second big law, his second big principle, right? So he had the principle of segregation, and then he had the principle of random segregation or independent assortment, right? So those were his 2 big principles. And that kind of set genetics up for the 20th century. However, there's one more probability rule that we have. That's the rule of multiplication. It seems very similar, but in this case, what happens is we have two different independent events, just like we did with the rule of addition. In this case, what we want is we want an event to happen in A and B. It's a different question, right? And remember, this is the word trigger for multiplication. So what you're asking for if you're flipping quarters is you're saying, what's the probability of flipping a heads in the left hand and in the right hand? You want both to happen, right? So that's a different ask now. You're not asking either or, you're asking this one and this one. So what's the probability there? Well, what it is, it's the product of their individual probabilities. So you multiply their individual probabilities together. So let's take a look at this. We're going to cross our two heterozygotes again. And what we want to do is we want to know what the probability of obtaining the homozygous recessive is. So what's the probability of getting the little p from dad? It's 50-50, right? So that's 1/2. And the probability of getting the little p from mom? Again, 50-50, right? So what's the probability of getting a little p from mom and a little p from dad? That's what you're asking, right? Well, in that case, it's going to be multiplying the two individual probabilities, which comes out to 1/4. And if you take a look at the Punnett square, it also draws it out for you, doesn't it? Here's your homozygous dominant, here's your heterozygote, here's your recessive. You have 1 in 4 options, right? So you have 4 outcomes. One of those is going to be the homozygous recessive. So you can get this one of two ways. You can either get this mathematically with the probability, or you can actually get this by just sketching out the Punnett square and counting boxes. Either one will get you your overall outcome. Okay, there's a couple of other things, though, that we want to kind of cover that are important in genetics. And because we kind of run into this, right? We ran into this, for instance, with Mendel, right? So you get this idea. So let's say you get this actually, no. Let's say you get this situation where you get this plant or whatever, and it's got the dominant phenotype, right? So the question is, what is that second allele? Do you know? You don't, right? Not in Mendel's system. So what you have to do is you have to test for it. So this could either be little a, or it could be big A, right? You don't know which one. So in a test cross, what you do is you basically take your unknown genotype and you cross it with a homozygous recessive. So this is what you do. You take your homozygous recessive, which you know will only give the little recessive allele, right? Little a. And this one will give either big A or little a. You do the same thing over here. There's your tester. This will give either big A or big A. So your offspring then will basically have a diagnostic look, right? In this case, you'll put a heterozygote, homozygous recessive, heterozygote, homozygous recessive. So now what you're going to have is a 1-to-1 phenotype, right? Half dominant, half recessive look. In this one, it's going to be heterozygous, 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 heterozygous. So it's going to be 100% 100% dominant. So in just one quick cross, you can determine what that allele is. Why do you use the recessive allele? Why not something else? Because the recessive allele, you know what that is. There's nothing else in there, right? So any difference between these two crosses is going to come from that unknown allele. Notice, you know all four of them. You know this one. You know this one. You know this one. This is the one that's a question. So by adding known alleles to the system, it flushes that one out. Okay, you can only say that for recessives. Okay. So it's kind of what it looks like. So here is your alternative one where you get all dominant versus a 1-to-1 ratio. So this is your heterozygous versus your homozygous dominant. We do this in genetics labs all the time still. Because sometimes we'll see this dominant, we're like, I don't know what that is. I need to know what genotype that is, right? Because what I need to do is I need to generate a true breeder. So I need to make sure that this guy is not a heterozygous, because otherwise that's going to screw all my results up. So you do a quick test cross and you can determine whether or not you've got a true breeder or not. So when you do a test cross, you can do it for a single gene like we saw before, or you can do it for double genes, a dihybrid, for instance. Like in this particular case, you can see that when you test these, A and B will breed true, right? Because they're both homozygous. In this case, B will breed true because it's homozygous, but A will not because it's heterozygous. You flip that around, B will not breed true because it's heterozygous, but A will because all it has is homozygosity. And this, none of them will breed true because they're both heterozygotes. So in a lot of ways, you can actually use a test cross like on organisms like this, where you have lots of different genotypes and you're just trying to figure out what they are. This is a really, really quick and dirty way to try to do a lot of genotypic analysis on some unknowns and to try to really figure out what you've got a hold of, especially if you've got a large population of unknown organisms with unknown genotypes. So to kind of inject a little bit of 20th century DNA, right? We now know, for instance, that genotype and phenotype are related, but we also know that the genes are going to be residing on DNA, and the DNA basically forms the basis of our genome, which is essentially a collection of genes. So that's what's actually in your genome, and a gene basically is a sequence of nucleic acids that dictates your amino acids, right? That's basically how a gene expresses itself. We'll talk more about this in a couple of chapters. Now, what happens if you mess up this sequence? Well, that's basically where you get a mutation. So you'll alter the identity of the DNA, which could alter the identity of your amino acid sequence, which could change the identity of the protein. Then, of course, ultimately, then it's this mutation or this alteration of DNA that can then be acted upon by natural selection that can then lead to evolution. Here's a problem, though. Oftentimes, a mutation is a major mechanism for evolution. But part of the problem with mutation is that, generally speaking, mutations are mostly negative. So, for instance, in order to have a favorable evolutionary progression, the idea is you have to accumulate some mutations that have a beneficial effect on you. And by and large, that is not the case. Most mutations being random is kind of like throwing a monkey wrench into an engine and hope that improves your engine, right? That almost never does. And so the amount of time it takes to accumulate beneficial mutations is actually quite long, right? Which is the reason why we're talking about billions of years, in terms of time frame, when it's like, I think 3 or 3.5, 4 billion or so, like when this first cell started coming together, right? Because you're talking about progression of speciation by mutation. It's a genetic idea, right? Evolution is a genetic idea. But the problem is mutation rates themselves kind of have a fundamental problem associated with them, especially when you take a look at evolution from a more of a classical Darwinian approach. It just takes a really, really long time to accumulate that many mutations that are favorable. Because if you take a look at a mutation rate, which is usually 1 in every 500 to a million years or so, I mean, it's really a long period of time. The beneficial mutation rate is even longer because the overwhelming majority of mutations are negative, which really puts a lot of strain on the classic Darwinian timelines, because then it's like, well, wait a minute. So this is actually taking more like 14 billion years if you use currently published mutation rates to actually accumulate the mutations that we would need in order to speciate to where we are. So the mutation rates are problematic. And this is honestly the reason why, and it's always has been, right? And part of the reason why is because we didn't begin the evolutionary discussion the way it should have begun, which was with genetics first. And we started it with a bunch of rock zoo stuff, right? We started it with the back end and tried to explain it with philosophy and all that kind of nonsense, right, ahead of time. When what we really needed was to wait patiently for genetics to come into its full flower so we could really appropriately discuss it. Now, this rub between sort of the mutational shortfalls, and also I would argue there were some pretty significant shortfalls in the fossil record, which is the reason why I'm not interested in the fossil record at all. Is the reason why in the 60s or 70s, I think, why Stephen Jay Gould and Elledge came up with punctuated equilibrium, right? Because fundamentally, it's like when you took a look at the classical Darwinian thing, it wasn't matching up. It's like the time frames weren't matching up, the progression wasn't matching up in some cases. And so what they discovered was that mutation rates don't support a Darwinian model by and large. However, what Gould and Elledge proposed was that for the most part, evolution goes in like fits and starts, right? So you have these long periods of equilibrium where Darwinian processes reign, but then every now and then they're punctuated by seasons of catastrophe and upheaval where mutation rates will change rapidly, and then they'll kind of alter. So they're not the slow creep that they used to be. The mutation rates get very fast over a very short period of time, and you get rapid speciation over a very, very short period of time. And so you kind of had this sort of punctuated, right? You have normal Darwinian progression punctuated by these spikes of rapid speciation, generally driven by increases in mutation rate. And so that was highly, highly controversial, especially coming from UC Davis, which is a Darwinian school. So you could imagine how well I fit there. But I mean, they had, so a lot of these ideas are kind of an amalgam. And so when you take a look at that, that's the reason why punctuated equilibrium came along, is because of the failings of the classic Darwinian model. And then there's other issues also with the Darwinian model on a geological time scale as well. Like, for instance, the geologist, which this is kind of like the temple classroom for geology. This is where Dr. Adams teaches out of. Right, but I mean, the classical geology only had, slow creep, slow process things called uniformitarianism, right? Those slow creep processes, which required billions of years to happen. But the problem is originally geological creep, those slow Darwinian processes, completely negated the role of catastrophe, right? So the idea was that everything is laid down by this glacial creep of sedimentation, right? Things like that, like leaving no room whatsoever for catastrophe. And that's part of the problem with early geology, is it was all very uniform rates and all very slow, but they refused to create room for catastrophe. And that was their failing, because we know now that the environment is anything but uniform, and it's actually prone to extremely violent fits of catastrophe, which can be quite earth altering. I mean, a good example was Mount St. Helens. Right, Mount St. Helens was a massive geological reconfiguration of that area in a very short period of time. And so now what you see is kind of like a grudging admission, granted, but an admission that, okay, yeah, we have the slow creep, but every now and then there's these episodic sort of massive catastrophe events. And so back in the day, the whole meteor thing, Chicxulub, for instance, was controversial because the idea was the dinosaurs died out with the Darwinian slow die-off. And there was no catastrophe. But now it's like, okay, there's mass extinction events. They happen periodically, right? Something bad happens, half the planet gets wiped out, we go through a bottleneck, and then we reemerge. And now we kind of have a better understanding. So what we understand now is that in reality, it's a mixture. There are some systems that are kind of Darwinian in nature, that they kind of are better explained by the slow creep, mutation rates, and things of that nature. And there are others that are better more explained, punctuated equilibrium and things of that nature. So we're slowly kind of getting to a little bit, although it's been very grudging, to kind of an acceptance that there's kind of an amalgam of different issues going on here, but there's still a long ways to go. Because even though a lot of practicing evolutionary biologists are kind of there, most of our textbooks are teaching evolution the way it was taught in the 19th century, which I balk at and I roll my eyes at. And that's my problem with evolution education. So there's that. Which is funny because when I argue in favor of genetics as the core of everything, including evolution, I always remind my evolutionary friends that it's like we have 6 major mechanisms of evolution. Most of them are genetic in nature. Like mutation is a genetic phenomenon, neutral drift is a genetic phenomenon, right? So most of these things are genetic in nature. And that kind of just goes to show you the importance of genetics, even within evolution's construct. Okay. So when Mendel basically put away his work and it was rediscovered, that's when the good stuff happened. So Mendel was kind of cool. We can kind of recapitulate his work. But now we want to do is we want to get into some of the extensions to Mendel. And so I want to kind of get a little into the middle of this here so I can bounce in the middle of some of this stuff and not hither and yon. I'm going to put it right about there. I'm going to put it right there. And I'm going to bounce a little bit. But I want to get started a list of some of the extensions to Mendel. So these are the things that we discovered after Mendel. So when we first discovered, or rather rediscovered Mendel in the 20th century, some of us found that what Mendel saw was what we saw. Right, so we would cross our organisms. We see the same 3-to-1 ratios. We'd see the 9-3-3-1 ratio, so it looked great. Others of us didn't see that. We saw something else. And so generally speaking, those individuals who saw something other than what Mendel saw usually had one of 2 responses. Either Mendel was wrong, right? Or maybe what's happening in my system is a little bit different. Let's go and dig into my system a little bit more because maybe there's a different set of rules here. Generally speaking, the people who were dismissive of Mendel and his work have been forgotten to history, which is a lesson to learn in science. So oftentimes when you dismiss another colleague like that, it's because you are being arrogant and foolish, and history will forget you. So that was the wrong response. The right response was, well, wait a minute, maybe it's doing something different in mind. Well, first of all, this is what's called a look at what we refer to as patterns of inheritance. I want to lay down what we've learned so far from Mendel. These are alternate patterns of inheritance. So first of all, the first rule, right? These are like rules by which genes will segregate. The first one is the Mendelian pattern. There's a couple of basic things. First of all, Mendel said there's one gene to one trait, right? There's only a flower gene, one. He also said that there's only 2 alleles. You have purple or white, tall or short. The other one, he said, there was complete dominance. What does that mean? That means when you're heterozygote, guess what? You only see what? The dominant. You don't see any white bleed through, right? There's no blending involved here. So those are three major things that Mendel saw. And then, of course, we're not going to look at this one, but he also looked at random assortment. Right? That everything was random and independent. Well, the first thing that we want to do is we want to take a look at variations of Mendel's idea. The first one we're going to take a look at is what's called polygenic inheritance. So polygenic inheritance is basically where there's many genes involved in one trait. So since we are at our witching hour, just about, I believe, I will start next time with this slide, polygenic inheritance. And we will basically fill out Mendel's list. And then each one of these, like in the genetics class, we kind of expand on and deepen and broaden, and we add other things to it. And we kind of add extra variables, right? So instead of like color by number in genetics, we actually just give you your own paints. We teach you how to mix the colors and create different shades and things of that nature. And so that's basically where I am going to start next time. Yes, I'm going to give them all back on Wednesday. I'm going to give people a chance to get back, because otherwise it's kind of, it goes out a little bit and then we kind of start nickel and diming. And then I don't know who's got what and where. So I want to try to get as big a crowd as I can, and then I'll give them all back to you guys. Remind me, though. You'll probably have to remind me because I'll probably forget. So we'll be in this room probably for the rest of the semester. So just